Where'd you go? There you are. Do I put the right one right now? Okay. Which one is it? That one.
Yeah.
All right.
We're good to go.
All right, everyone. Welcome. I'm rather impressed to see you in such a high number on a Friday evening. I sense a lot of commitment. I'm very impressed. So thank you all. Tonight, actually, I should say, on this Friday evening, tonight we're going to be trying to talk to you a little bit on how we think we begin to have a beginning of an understanding of how the brain works. As you know, this is an event that was organized by the Brain and Mind Research Institute at the University of Ottawa that lasted the entire week. We've had evenings on dementia, stroke, Parkinson, and which was really to showcase the research that is being carried out right here at the University of Ottawa under the auspices of the Brain and Mind Research Institute, which brings together roughly about close to 100 labs working on different aspects of brain health, of brain function in health and disease, distributed over several faculties at the university. And we all aim to work together in order to essentially decode and understand what is a very, very difficult puzzle. And tonight, so we're going to be talking to you about how we're trying to decode the brain, and essentially what we're trying to do is crack the neural code. In that, how is it that the brain can perceive and store memory, and how does it modulate mood, and how do these mood ultimately influence our daily activities, our decision-making processes, and ultimately how does the brain, how do thoughts emerge from brain function? So to do this, so I will begin. So my name is Jean-Claude Béïque. I'm a professor here at the Faculty of Medicine. I will kind of give you a bit of a Neuroscience 101 class. And then we're going to go in a little bit more details. And then, André Longtin is, so you're going to go, no, I thought, okay, so who's going to go? Okay. So Richard Naud, who's also a professor here in the faculty, is going to talk to you a bit more about some computational work that he's doing in order to understand brain. And then we're going to have Emerson Harkin. Emerson is a PhD student. He just finished his master's degree in my lab in collaboration with Dr. Naud. And now he's just undertaking a junior PhD student in the lab of Dr. Naud, and is going to come and talk to you guys a little bit about his work, and in order to hopefully give you a sense of how research is carried out in university setting, which is, in a nutshell, is university professors, they don't do much. All the work, all the real work is being carried out by the students. So Emerson is going to come and talk to you about it a little bit more, and then we're going to finish with André Longtin, who's also a professor here in the Faculty of Medicine, but he's also in the Department of Physics. So without further ado, we'll try we're going to go in the crux of the thing. So I want to begin by asking you a simple question. This is, why do we learn? We have a pretty good idea that we get a good intuition that our brain has this fantastic ability to store information and we can recall it. So why is it that we need to do this? And in order to answer this question, I want to give you some sort of a bit of an evolutionary perspective of this. And in a nutshell, we have this amazing ability to learn because it's evolutionary advantageous. Now, so think of a bacteria. Bacteria has been around for a long time, for like 3,500 million years or so. They're still around. So that means that they're doing something right. And that's one of the hallmark of evolution, is that if you're doing something right, well, you're going to stick around for a long time. Now, when we think about evolution, there's a number of features that are super important. One of them is when evolution finds something that works, it keeps it. So you guys know about DNA. You know about it makes proteins. And then I just want to show you this, ATP, ATP synthase. That's an enzyme that takes ATP. And maybe some of you have already a good understanding that ATP rhymes with energy. Everything, when any biological system needs energy, it gets this energy from ATP synthase. Now, the ATP synthase that are in bacteria and the ones that are in your body are exactly the same. Every single living organism in the kingdom has ATP synthase. And it's been there forever. When nature finds something that works, it keeps it forever. Okay. Now, no disrespect to bacteria, but in order for a bacteria to survive throughout millions of years of evolution, what it needs to do, it needs to interact with this environment, but it doesn't need to do it in a very, very complicated way. But still, bacteria learn to interact with their environment. If you change nutrients level, they're going to induce some other enzymes in order to capitalize or to optimally interact with their environment. And so bacteria will do this, but you can get a really good intuition that as you go through evolution, the level of interaction with your environment gets ever more complicated. Now, and this is the trick. Your environment is dynamic. It changes all the time. You need to be able to interact with it in an optimal way. That's the only way that you're going to survive. And that's the only way that a species will survive through evolution. And with that learning, there's absolutely no way that you're going to be able to do this. And if you take this all the way up to humans, well, it's easy to imagine that a number of aspects of learning gives you evolutionary advantage. Even things like as abstract as, say, for example, language. If your brain is able to compute and able to learn a language, then if you're in a tribe, in an environment of limited resources, if you're able to communicate with one another in a meaningful way, well, your society in general is going to, or your tribe in general is going to be in a better position to interact meaningfully with this environment, and you're going to have a competitive edge. Right? This is how evolution essentially puts pressure for you to have ever more sophisticated learning abilities. And with this as a backdrop, now you can get an understanding that learning is important, and it is, and has been for a long time, and it still is, amongst the most pressing and grand challenges of modern neuroscience research. We're trying to understand how does the brain is able to store information and recall them. And part of the reason is for evolutionary reason. And you can all get a good intuition in how you need to interact with your environment to be able to adapt to it. Right? If you put your hand on the stove, hopefully that you'll do it only once. Right? Okay. So now we're going to get into more of the crux of it. So obviously, if you want to study how memory works, you may want to study this in humans. And enters here the single most famous case of cognitive psychology. And this is the case of Henry Malaysian, also known as HM, who was a patient, and around the age of 7 or so, he had a bicycle accident. And from then on, he suffered from epilepsy. And sometimes you suffer from epilepsy, and it's intractable epilepsy. That means you're taking drugs, and they do not work. And it's obviously a highly debilitating disease. And one final treatment for epileptic seizure, epileptic patients, is you actually go through surgical resection of the brain region that is responsible to initiate the epileptic seizure. So HM undergone, I think it was in Connecticut, undergone one of the surgery where he removed parts of the temporal lobe. And in removing the temporal lobe, he removed a number of structures, including the hippocampus, the amygdala, but mainly the hippocampus and entorhinal cortex. So, and luckily for him, it treated his epilepsy. However, he had a number of consequences, we could say, for his daily lives. And Brenda Milner, who was then a PhD student at McGill University working with Wilder Penfield, decided to study the HM. And HM had a very unique, very unique clinical presentation. For one, he had retrograde amnesia. That is, for a couple of years before he had essentially no memory for a couple of years before his surgery. He could draw a plan of his house in which he grew, but Brenda Milner used to see him every single day, and every single morning she had to reintroduce herself. He could not store any new information. So you remove his hippocampus, and he cannot make any new information. Now, and she did like a really clever experiment that's going to be it's very simple, but in a way opened up an entire new field of memory research, which is this experiment here. So you come in, and everybody here would do this, and you would come in a lab one morning, and somebody would ask you to draw a star. But you cannot see your hands. You can only see your hands through a mirror. And it's a bit tricky, right? So you're going to start to draw your star, and you're going to make a whole bunch of mistakes. And these mistakes, it's easy to quantify. And then you can label this in terms of errors. So in day one, you start to do the experiment, you make a whole bunch of mistakes, but you adapt to it, right? And then so you're getting better and better and better at this, such that by day 3, your level of errors is pretty low. So clearly, when you're doing this, your brain is adapting with its environment, and it's changing. You are learning how to do this particular task. So she did this experiment with HM, and every morning when during those days, when she was doing these experiments with HM, and this is one of the many, many experiments that she did, HM did not remember how to do the experiment. Right? So she had to re-explain to him, you got to draw a star, blah, 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 through this mirror, and so on. I said, okay. But when she did that experiment, she realized, and that was a huge eureka moment for her, wait a second, he's getting better at this. He could not remember it, but he was getting better at it. So his brain was learning how to get better at it, but he had absolutely no recollection. And this was really the sort of like the foundation of a modern understanding of memory. That's memory. There's different types of memory in the brain. And this is procedural memory. This is one of them. You're playing ping pong. Or episodic memory. I remember from the wedding or your own wedding, or the wedding of your children. These are fundamentally processed in the brain in fundamentally different ways. Okay. So this is sort of a nutshell of a snippet of how we understand memory processing in the brain. And you could do this now these experiments were done more than 50 years ago. And obviously it's becoming more and more sophisticated, for instance, with brain imaging techniques that allows you with remarkable precision now to observe different brain regions that gets turned on and off during everyday tasks and, for instance, during memory processing. So this is fantastic, but there is a fundamental limit to what you could do with humans. For instance, I'm an electrophysiologist, so I put electrodes in neurons. This is what I do for a living. I cannot do this in your brains. My dean won't let me. Right? So enters here, this one I want to give you a sense of daily activities in labs. So we do this in animal. We do this, for instance, in transgenic mice. There's a number of animals you could do this in rats, in monkeys. We don't do it here. We could do it also in simple organisms, such as Drosophila and different types of worms, that they have a whole bunch of genetic advantages. Now, so if we're going to talk about animal research and modern neuroscience, we have to talk about these guys. We have to talk about Camillo Golgi, who developed a technique to see neurons. And we have to talk about Ramon y Cajal, who is still considered to be the father of modern neuroscience. And so these two did not work together, and actually, so they both won a Nobel Prize in 1906. They did not work together, and actually they hated one another. And when they got the Nobel Prize together, when they're on the podium in the front, they didn't even shake their hands. This is how bad it was. But he essentially was a neuroanatomist. And I'm just going to go through slowly with you and show you some of the pictures of the drawings that he did more than 100 years ago. And because they're still the drawings that we show our students now, because they're accurate. Right? So here you have, for example, whoops, I got too fast. So here, these are neurons that are found in your retina. This is a complicated structure. The retina is a wonder of sophistication. It can extract features of your visual space in insanely complicated ways through these highly, highly organized neuronal networks. This is Purkinje cell in your cerebellum involved in coordination. These are amongst the biggest neurons in the brain, and they have absolutely fantastic dendritic arborization. This is the hippocampus. This is the hippocampus. This is what was removed in HM. You need this to store memory. You need these complicated neurons that are making these highly, highly processed networks. This is cortex. And this is one single neuron. This is a layer 5 pyramidal neuron in all of its glory. A single typical neuron in the brain of cortex will receive about 30,000 synapses or so. That means there's 30,000 contacts on that one neuron with other neurons. And every single one of those synapses, there's a computational unit. And I'll come back to that in a second. Now, if we're going to talk about brain, I just want to give you an idea of the scale. So this is a human skull here is about 15 centimeters or so in diameter. All the white stuff here, this is white matter. See this as electrical wires. When the back of your brain needs to communicate with the front of your brain and other networks, it goes through these electrical wires, which is white. All the gray part around, that's your cortex, is where you have all neurons. And so if you take you increase this part here, 50 micron here. 50 micron, that's human hair is about 50 micron in diameter. It's packed with neurons. And then if you go even smaller, that's a piece of dendrite right here. All these little spines here, they're little protrusion, they're called spines. That's one synapse. So you have about 30,000 or so for every single neuron in cortex. So I'm going to just give you a bit more, just a bit of a visual representation. And here you have all these neurons. So there's a huge problem in the brain. It's called real estate. You need to pack stuff. And then here, all these are different, color-coded, different processes from different neurons. All right. So told you there's 30,000 synapses. So how does it work? Well, sorry. In a nutshell, here there's a neuron here, and then here that neuron, that's an axon coming from a neuron that could be in a completely other different part of the brain, fires an action potential, it travels down, and it makes a contact. And this axon here will release neurotransmitters that it's going to go and cross and activate the other neuron. And then it does so by filling all these vesicles with neurotransmitters. And that's at a single synapse, and this is how you communicate. With one neuron communicate with another neuron by single synapse communication. What is the nature of that communication? That's hard. That's a hard problem, and we're still figuring it out. For instance, Richard and Andre and myself, we're trying to figure out what is the amount of information that's being transferred from one synapse, from one neuron to the other at the level of these synapses. Now, one way that we do this, just to give you a bit of an idea of how we work in the lab. So students come in the morning and they make slices of brain. And one of the things that we do is that we use a lot of optical approach because it's easy to manipulate light. So for instance, this is one neuron, and this neuron, contrary to the neurons that I've showed you from Ramonica, this neuron is alive. So you can obtain recordings, and these are little synapses that I've showed you, and you can shine laser and activate a single synapse, and then these are tiny, tiny, tiny different currents. In this case, it's 5 picoamps, and this is how the brain uses electricity. It doesn't use electricity to make a movement. Doesn't use electricity for mechanical force. It uses electricity to communicate. That is the essence, the bare bones of the neural code. That's what we're trying to figure out. And these neurons are, so this is one of these pyramidal neurons in all of their glory. At their core, they're input-output machines. They're computational units. This is what they do. They receive a whole bunch of inputs, and then they make a decision. They take all these inputs, make a decision, they make a transform. That's the neural code. That's the essence of how we compute and how you generate thoughts. And so, for instance, here we're trying to figure out at a single synapse, we're imaging, we can image with special proteins, we can see neurotransmitter being released or not at single synapse. How are they released? What's their dynamics in space? What's their dynamic in time? Again, trying to crack at the most fundamental aspect of what's the nature of information being transferred. And then this is a number of other examples obtained from the students in the lab, and they can change with and try to interrogate really the mathematical operations that are being carried out in these dendrites. And then you can generate computational models where you can put meanings to what you're finding. You could put words, they segregate, they amplify, they attenuate. These are the operations that are ongoing in your brain constantly as I talk to you, but we're trying to understand how this thing is occurring. Now, we have to talk about plasticity. So what is plasticity? You probably heard about synaptic plasticity. It's a word that's being used over and over and over. So I'm just going to read this, and because this is pretty much how we view synaptic plasticity. All right, so one can admit as very likely that mental activity, so you have the process of mental activity, provokes a greater development of the nerve collaterals and the part of the brain most utilized. So say if I repeat my phone number to you guys a number of times, you're going to be using your brain in some ways, right? You get the concept of activity, and then that activity is going to do something to your brain. Maybe you're going to have multiplication of the terminal branches and nerve collaterals, or you can have novel intercellular connection. These are synapses. You may have more synapses. So the concept that you have activity that's changing something to your brain, that's synaptic plasticity. And that was formulated more than 100 years ago. By and large, this is still correct. So I'm going to give you one example of a synaptic plasticity experiment that was done in the lab. So you have here, these are all the thousands, one of the thousands of individual synapses. And then with these lasers, you can activate these synapses repeatedly. And then this, so you can follow this over time. You can follow this synapse and see what happens. It gets bigger following activity. So this is how we view synaptic plasticity. So when you're working, say, at home on your computer, on your Word document, and then you press save, there's a fundamental change. There's a physical change on your hard drive. You press save of on your computer, your hard drive has changed. Right? How does that generate a song? How does that generate a Word document? You don't know. but you know there's something that has changed on your hard drive. This here, that's a change. That's experience provokes activity, synaptic activity provoked a physical change. So we're pretty good at this. This is where the field is right now. We're pretty good at seeing and understanding how this change occurs. Molecular mechanism of this, cellular mechanism of this, we're pretty good. But still, how does, if I show you with a microscope a hard drive, and I show you, oh, there's bits that have changed on that hard drive, how does that generate your song that you just stored on your computer? That's a hard question. And that pretty much sums up the challenge that modern neuroscience is facing right now. We know about synaptic plasticity. How does that relate to a memory? That is starting to get a bit harder. And I'm just going to end this, and I only have a few slides left, with one of the theory where we think we can change, where we're linking these changes at the cellular level with an actual memory. So there's this thing called the grandmother cell theory. And it's been around for a long time. And the idea is that when you see your grandmother, there's a neuron in your brain that spikes, that starts to fire action potential, and then, hey, this is grandma. Turns out that it's probably not that simple. Now, I'll tell you the current theory of memory. And it's not that hard to grasp, to get a good intuition. And so imagine that you have a set of neurons that are all interconnected. And you present these neurons over and over and over some external stimulus. In this case, it could be your grandmother. So you show your grandmother over and over and over, and you have all these neurons that are being activated when you see your grandmother. And then they're going to make synaptic plasticity because they're firing together all the time. These networks are going to get, these synapses are going to get stronger. And then you're making a cell assembly of your grandmother. Now, once you have this, now what happens if you see only a partial representation of that external stimulus? So what if you see your grandmother in the dark? You're pretty good to see, this is grandma in the dark. You don't see, or you hear her voice, right? That's called pattern completion. This is how we think that the brain stores information. So there's a bunch of cells that are representing your grandmother, and if you can activate only a few of them, you complete the pattern. This is grandma. And your grandmother may be really nice to you, and then maybe that pattern completion that you're going to complete in parts of your brain of mood-related networks in your brain that say, oh, this is good. I get this good tummy feeling. I'm happy because my grandmother used to cook chicken noodle soup when I was, and then, right? And the classic, most of you have heard of the infamous how do you call this? The French, I'm blanking out now. That little pastry, that French pastry. Les Madeleines de Proust, yeah. Right, so most of you have heard about les Madeleines de Proust, where Proust, he just smells, Madeleine are a little pastry, and he smells the pastry, and then he has massive recollection of his childhood memory, right? This is pattern completion, right? So it turns out that we don't have a lot of evidence for a grandmother cell, but we do have experimental evidence of a Jennifer Aniston cell. These are real experiments that were done, and they were presenting to an individual where he was undergoing surgery, and you were presenting a number of individuals, and you had one cell that was, you had blonde person, blonde males, blonde females, and the neuron wasn't responding, but when Jennifer Aniston was there, that neuron was responding every single time. Right? So it was probably one of the cells that was doing pattern completion. And then I'll end for this. One of the things that we do in the lab now is we're trying to figure out how mood, different mood, this is from one of the Pixar movies, and it turns out that it's actually fairly accurate, where you have all these networks, distributed networks, that are encoding these types of things, of fear, sadness, joy, anger. And these things are all encoded in different parts of the brain, and we're trying to nail down how are these encoding these different types of emotions, and how do they contribute to diseases such as anxiety disorders and major depression. And real way how we do this, again, we use light. Because light is we can now, with modern techniques, we can manipulate individual brain function and individual brain activity with lasers and a whole bunch of light. And this is just one example. This is a neuron that's spiking. You shine light, and then you can activate it through protein engineering. And then you can take these animals and activate these specific types of neurons with light and see how this influences individual neuron function, individual network function, and all the way to the level of behavior. Okay? So that's all for me. So I'm going to pass the baton to Michel.
All right, do you hear me well? Perfect. So I won't do as Jean-Claude told you I will do. I decided to talk about AI and the link between artificial intelligence and neuroscience. I decided that this morning. I thought it is a topic that interests you and it's related to my work. So I prepared my lecture in that direction. So, there is a lot of things conspicuously missing from this. And interestingly, they're here, so. Yeah. And we tested it earlier on.
Right, do you want to just go from the
Well, AI doesn't really help you for this.
Do you want to just go to the original?
Yeah.
Online, oddly enough. I have the same slide, but with like this pinboard and a bunch
Right.
of stringly things.
Yeah,
That's what's supposed to be on screen.
that's correct.
So since I got this on the
Yeah, without people like Victoria, I'd be lost. Thank you. So, let's start with this. So, let's go back in time to the birth of the time before artificial intelligence, before computers really, in some ways, or modern day computers. There's Frank Rosenblatt, who had this machine, this complicated machine that he called a perceptron. And he was studying that machine feverishly, and he said, well, my perceptron is not primarily concerned with the invention of devices called artificial intelligence, but rather with investigating physical structures of neurodynamic principles which underlie natural intelligence. So at this very onset, you had people that were studying something like artificial intelligence, but really to try to understand the brain. And it's from this work that we now have things like this, self-driving cars. The work that Frank Rosenblatt started evolved into something else. And now we have a machine that is able to look at an image and recognize when there's a pedestrian that's hiding behind a car and it's going to appear on the other side. This is truly marvelous. I still can't believe that we've managed to figure that out. And you might think that the machine is just a machine and it has nothing to do with what the brain does. But there are surprising parallels, and that's what I want to talk about today. I want to ask the question, how do brain recognize the content of visual stimulus? And how does artificial intelligence does the same thing? So at the beginning of the problem, both the computer and the humans have a similar thing. All they see, say, the computer here has an image that's really pixels, right? If you blow up an image, just pixels. And what are pixels? Well, it's just squares where we know the color or the light intensity. And so it's just numbers. It's just numbers on the grid. How does the computer go from these numbers on the grid to recognizing the content of an image, saying there's an animal? We have the same problem. In the retina, light comes through the eye. It goes to the back of the eye where the retina is. And there are the photoreceptors. And they translate the light energy into an electrical energy. But it's just electrical energy on a grid. How does the brain then build on this information to extract the meaning of the image? One of the first to ask this question is Stephen Kuffler. And he wanted to know, well, what does the retina tell the brain? So Jean-Claude told you that the retina is a very complicated array of cells, so it can do a lot of things. Maybe the retina is actually able to process information in a way that it can recognize a cat and tell the brain cat. Is that what happens? So he wanted to test that question, and to ask this, he did experiments with an electrode at the back of a retina of an animal. And he recorded action potentials as he was shining light on the retina. So he wanted to know, well, if I put my electrode somewhere, and then I shine light somewhere in this visual field, will I see a response? When will I see a response? What type of visual stimulus is being communicated to the brain? So he put the light someplace, no response. Put the light somewhere else, closer to where the electrode is. A response. Aha. The retina is sensitive to light, but only in a particular spot. If I put it somewhere else, it won't respond. If I would have put the electrode somewhere else, well, it would respond to maybe that particular location. Okay, what happens if I make the spot bigger? No response. Well, that's kind of strange. And he played around with this, and he realized that the way to get the maximum response is to surround this spot with a halo of darkness. This is just very strange, and there's kind of not much you can say about this. But all he concluded, and what you can conclude is, well, all right, what the retina loves, because if I find a neuron in the retina, the stimulus that gives a maximum response is this spot with a shadow. So retinas, the cells in the retina, they love spots with shadows. Okay. Now, what happens further down the road? So if information from the retina travels to the back of the brain, and where we have a visual cortex, what about cells there? What do they love? What type of visual stimulus do they love? So this question was asked by Hubel and Wiesel in 1962, won the Nobel Prize for this. And they were showing visual stimulus on the screen while recording from the visual cortex of a cat.
The researchers actually listened in to individual nerve cells firing in the anesthetized cat.
I didn't want to show, I don't know why the video was not the same as the one I had on the... This is the love of changing between formats. So, they were playing with the, this is them recreating the experiment that they did in 1962. And they were trying to find what sort of stimulus can I show that will drive the maximal response. And they were trying with those spots because, well, that's what worked for the retina. So maybe it would make these cells respond. And it was not quite working. Or at least they were finding.
Black dot seemed to be working in a...
The crackling sound is that they've connected the electrode to a loudspeaker. So you hear the neuron that they record in the brain. And the response was kind of strange until they realized it was by moving the slide edge across the screen that was driving the response.
Understand until we found that it was the process of slipping the piece of glass into the projector, which swept a line, a very faint.
And from this, they realized, well, if I slide this bar of this edge of darkness or edge of light with darkness in one direction, I get no response. But if I change the direction ever so slightly, there's a particular direction where that elicits a maximum response. And this is thus visual stimulus that these cells love the best. They concluded that cells in this visual cortex, they love these oriented bars. So this didn't quite, it's very interesting and fundamental somehow. We've understood a little bit better what the cell's activity loves from the visual stimulus. But how can you make sense of all this? But Frank Rosenblatt was saying, well, my machine does all this. It's all right. It's all simple. You're saying, well, it's easy. You can see neurons like a light bulb. Okay, you can imagine this. Let's follow that analogy, because if I put some light in one spot, and you can imagine that there's a connection between that particular spot of the image to, say, a positive connection on your light bulb, and you will light up your neuron a little bit. If you make this spot a little bit bigger, you can imagine that the surround of this spot is connected to a negative connection on your light bulb. And that actually reduces the response, so the neuron doesn't respond. And if I put a halo of darkness, then you kind of have this double negative, and it makes the light bulb light more. I can also explain to you why you have a bar, but why the neurons in the visual cortex love the bars. It's because if you have a different arrangement of connections, if you have connections to the plus with connected to in a line that is you following your preferred orientation, then you just accumulate power to your light bulb. Whereas if you have a different direction, you might imagine that the one that's offline will bring in some inhibition and prevent the response. So all this to say that you can just wire things differently and connect this to a response, and you can recover that sort of selectivity. So Frank Rosenblatt was like, I've figured it out. I can just now wire things up, and I'll be able to do very complicated things.
In the 1950s and 60s, scientists...
He was in 1960, however, and that was a little bit problematic for him. So he had these big computers that were in the time taking a whole room, and what he wanted to do is prove the world that his little machine made with wires was able to classify an image for being male or female. So they were showing these pictures to the machine, and they were saying whether the machine was trying to predict man or woman, in case woman, and then the experimenter would say it wrong or right, and then the machine would change the connections to try to best guess in the next time. It turned out that didn't work so well. In fact, it didn't work well at all. And they could not show that this was conclusive. But one particular thing about Frank Rosenblatt's setup is that it was an array of units connected to another array of units and another array of units. It was only in layers like this, and there were only 2 layers. And he might have thought about accumulating more layers, but he was limited by the computers at the time. The brain is different. Brain keeps on processing information. So this is your visual cortex at the back of the brain. And from there, information keeps on going, and it keeps on being processed. It goes through more layers of neurons. So what happens at the end here, the AIT, I'll call that the temporal cortex. Well, these is where the grandmother cells that Jean-Claude was talking about are lying. So this is an example not of a grandmother cell, not of a Jennifer Aniston cell, but it was recorded in if you record from a cell here, And you showed the person, here a patient that was undergoing surgery, a picture of Keanu Reeves, and these are spikes, these are electrical activity, and this is the total number of activity that you've seen in multiple trials where you've shown the same image. Keanu Reeves doesn't appear to drive any response, so this cell doesn't like Keanu Reeves. You can play the word Keanu Reeves. The cell doesn't like the word either. You can show written words of various things, Tower of Pisa here, and they tried a lot of things until they showed the face of this guy. And there was a lot of response. The cell likes this actor. It also likes written Luke Skywalker. It also liked the spoken words, Luke Skywalker. So this particular cell is really selective to a very abstract concept. And from this, you can imagine that what's happening in visual processing is you have an array of inputs, the light on the retina, RGC here it stands for the retina. And information goes through multiple layers of neurons. And with the change with interconnections, with a particular type of interconnections, you can get so that at the end, you have a grandmother cell. You're able to be sensitive selectively to a concept. So from cells liking spots with shadows, cells liking oriented bars, you get to cells loving concepts. So this work in neuroscience really inspired AI, because they were like, well, maybe Frank Rosenblatt was actually right. But what we needed to do is go from only 2 layers to multiple layers. So they thought, okay, we'll create a computer that has units in layers. And there's a lot of layers. Here they made like 300. And then we'll train this computer to recognize images. And maybe we'll have more processing power. And it's with this sort of technology, this sort of artificial neural network, that you're able to recognize the content of images. Now that's what's particularly interesting with this, and that's where I really wanted to go today with this story, is that you can ask, well, there are units in here. They're artificial units. They're not neurons, but we can ask a similar type of question that we've been asking the nervous system. What are the units here liking? What sort of visual stimulus do these cells like? We've trained this whole thing to recognize that here it would tell you whether it's a cat or a car that you see as an image that enters the entry here. But what are these units selective to? What are the stimulus that they like? Well, it turns out that the first artificial layer loves things like spots with shadows. That somewhere more in the middle, you have cells that prefer oriented bars. And only at the very top, you get cells that loves concepts like car or cat. So here's a surprising parallel with the processing of visual information in the brain. And what's more with this is that very recently this year, people have then used this to try to predict the activity of cells in the middle of the visual cortex of monkeys with surprising accuracy, much better than any neuroscientist has been able to do. I would have shown you that particular example, but I wanted to show this video that I saw today from a lab in France that's just made the news to illustrate the type of application we get from the work that we're doing here and trying to understand how the brain represents information. This man has been paralyzed for 4 years, and they've implanted the electrodes in his motor cortex on both sides, quite an array of electrodes. And he's been, even though the brain has never seen electrodes in it, it's learning to be able to use its own activity to control the electrodes that are in the motor cortex. And so that the man has been able to control robotic exoskeleton, and he's been able to walk. And this highlights both a few aspects of the type of work that we do in the lab, because we're really interested in trying to understand how, what's the language of the brain, how the cells are communicating information so that we can read it, so that we can provide that sort of capabilities. But also we want to understand how our brains are able to wire together. We know how artificial intelligence does it. It has very little to do with what neurons are doing, and as far as we can see, but maybe there's some inspiration there. That's the type of multidisciplinary work that we're doing here. And we also want to know why brains are so much more energy efficient than the machines. And this, by the way, my book on the subject. So with this, I want to let the stage to those to Emerson that is working in my lab and will really illustrate the type of work that is really being done. Thank you.
Well, hello everyone. Can you hear me okay? Yep. Great. I'm not used to using these microphones. So thank you for the introduction and the wonderful talks so far this evening. My name is Emerson. I'm a PhD student here at the Faculty of Medicine. But today I'm going to be talking a little bit about some of the work that I did with both Jean-Claude and Richard during my master's. So to try to kind of recap, a little bit some of the key concepts that we've heard so far this evening. We've heard that, essentially from the perspective of neuroscience, the mind is kind of what the brain does. We've heard that, at least at the level of single neurons, information is being represented in the brain by electricity. And finally, just from Richard, we heard that when you wire a bunch of neurons together, you can get them to do really interesting things, whether those are artificial neurons that are living in a computer, recognizing cars or pedestrians, or whether those are real neurons in a brain that are trying to recognize some kind of a visual stimulus. So with that in mind, I'm going to try to talk about how we can start to put a few of these concepts together to try to better understand a neural system that's kind of close to my heart, which is the serotonin system. So my talk is going to be, hopefully about 5 minutes long. I'm going to try to give you a bird's eye view of some work for my master's on reverse engineering the serotonin system. So the serotonin system has always been kind of close to my heart, because when we talk about this idea that the mind is what the brain does, from my perspective, when you have a system like the serotonin system that's so important for regulating mood, in a way, to reverse engineer the serotonin system is to reverse engineer mood, which when I entered Jean-Claude's lab a few years ago, I just thought was a really cool idea. So to give you a quick kind of introduction to roughly what we think the serotonin system does, the serotonin system is actually composed of a very small number of neurons located in your brainstem that, in spite of their small numbers, seem to have a huge impact on regulating a wide range of animal behaviors and perceptions. So those cells are involved in whether you're feeling hungry or full, whether you're feeling tired or really full of energy, or indeed whether you're feeling happy or sad. And one of the cool things about this system is that evolutionarily, it's a really, really ancient system. So as Jean-Claude kind of said earlier, If you see something that's really old in biology, often it means it's doing something right or it's doing something important. So clearly this system is doing something really fundamental in many different types of animals, including humans and mice and fish. All different kinds of animals seems to be doing roughly the same thing. So if you put yourself in the shoes of a mouse, just very briefly, since we often work in mice in basic research for reasons that I'll get to, I think, in a slide or two. If you put yourself in the shoes of a mouse, walking around, you might be thinking a little bit about, you might be thinking about where your next piece of cheese is going to come from. You might be worried about whether there's a scary cat around the corner. And all of this information kind of seems to be sent from a bunch of different parts of the brain towards this small number of serotonin neurons located in the brainstem. So those serotonin neurons, together with a bunch of inhibitory neurons that live in the same kind of neighborhood, somehow seem to assemble all of these different sources of information from all over the brain and use them to send a signal to the rest of the brain to trigger essentially the appropriate behavioral response. So, that's a little bit abstract, but what I really wanted to focus on for my project was that, we realized that now in 2019, we know a decent amount about what these inputs are, and we know a little bit about what kind of behaviors the serotonin system is regulating, but we really don't know hardly, very much at all about what's going on here in the middle. So this was kind of the driving question for my master's thesis. So almost from the start, this was a really interdisciplinary project, working together with Jean-Claude's group. I made measurements, basically, of the electrical properties of serotonin neurons and the other major cell types in the region where they live. So you can see me kind of buried here behind some microscopes collecting data. And one of the things that's really amazing about the Faculty of Medicine here is that you can walk right across the hall and talk to physicists and mathematicians in Dr. Naud's lab to generate computer models of this data. So in a nutshell, with a couple of years of work, we eventually figured out that this system is behaving in many ways a lot like a radio. So I put for my title here, how is a neural system like a radio? It sounds like a start of a joke, but it's actually, I think, hopefully it's a good analogy. So this is the system that we kind of started out with, and here's our fancy computer model of it. So there's a few kind of not so superficial similarities. Both of these are getting some kind of complicated, messy input. So for a radio, it's getting radio waves from maybe all over the world. In the case of the serotonin system, you're getting all kinds of information about different things in the environment from all over the brain. Out of it, you get a really clear signal, which is maybe your favorite radio program, or the direction to enjoy the little piece of cheese that you found or to run away from the cat. And there's some stuff that happens in between. So in particular for a radio, you've kind of got two different knobs. You've got one that selects the radio station. So in this way, you're kind of picking which input it is that you want to pay attention to. And some work that's been done very recently in Jean-Claude's lab that I won't speak about has uncovered a mechanism to do something like this in the serotonin system. And the other major knob is, of course, volume. So you can make your signal that you're picking up louder or quieter and kind of more or less distinct by changing the volume. And in a nutshell, by characterizing the electrical properties of these two groups of cells, we think that the interaction between them has some very specific properties that allows it to behave like an unsuspected volume knob on the serotonin system. So why do we think this is actually interesting or important? I think personally this is just kind of cool. I think it's interesting to wonder how these sorts of mechanisms can contribute to mood. But of course we can also apply this for the benefit of human health. So the serotonin system is really well known for its role in regulating mood. It's the target of most antidepressant drugs. But unfortunately, modern antidepressants don't really work so well. So what we kind of hope is that by understanding a little bit better these sorts of mechanisms, we can eventually in the future design better therapeutics to treat depression and other mood disorders. So with that, I wanted to thank everyone who's contributed to this work. So obviously the supervision and resources of Dr. Jean-Claude Béïque and Dr. Naud, who we've heard from. But I also want to thank all of our sources of funding, because this work really doesn't happen without access to funds to buy equipment and to pay our salaries. And when I say I'm thanking our funding agencies, really I'm thanking all of you, the Canadian public who pay the taxes that give these agencies money and are actually interested in this work. So thank you all for your time and for your interest.
One more to go. So thank you for staying on. Thanks, Emerson, for this wonderful talk. So I'm going to give you another perspective on the kind of work that we do here. And I am trained as a physicist, so I actually run the physics department here at the University of Ottawa. I'm the chair. But I'm also in the faculty of medicine, so I've collaborated with the neuroscientists most of my career. So I actually do both things, and you'll get a flavor of both topics in my talk. And I want to give you the notion also, there's this collective of researchers around the planet that have this kind of hybrid training that are coming at the brain with this novel perspective. The brain is a large area. There's about 70,000 neuroscientists on the planet. 5% of them might be interested in what I do. Another 5% might be interested in what Jean-Claude does. Oh, maybe no, 90% are interested in what. Okay. But we just have. It's a large. There's many different approaches to and many different questions. So I'm going to give you an idea of a few questions that I'm interested in. Okay. And they kind of sit between what Jean-Claude and Richard and Emerson were talking about. So now, okay. Let me just see if there's some, yeah, let's go back to the, I have it on the memory key too, but let's just pop it, get rid of that. Okay, here we go. All right, we're back in business, sorry about that. Okay, so the brain. I'm going to be looking at circuits, specific circuits, and I want to understand how the brain does specific things, computations. Not so much memory. I mean, I do some memory research with Jean-Claude and other people, but some specific things, like how do we count? How do we correct when the lights go up and we kind of adjust our contrast and so on. Various things like that. And like Jean-Claude was saying, we've been successful, evolving over millions of years. And a lot of that is due to the fact that some solutions that ancient creatures found to problems were propagated forward. And we are kind of a collection of all those solutions. And if we understand them in simple creatures, then it could help us understand them in us, that are very complicated creatures. Okay? Because our brain is much more complicated. There's many more cells, many more cell types. So we're trying to understand these circuits inside the brain. And when I talk to physicists, what we're trying to do is find the laws of physics for the brain, right? It's like, are there such laws? And so one thing that you heard about is magnetic resonance imaging, right? That gives you super clear pictures of in 3D what's going on in the brain. And it can tell you things, for example, what part of the brain is consuming more oxygen when I'm looking or when I'm talking or when I'm counting or meditating. It basically labels. It says, okay, this is what's important when you're meditating. This is what's important when you're talking. but it doesn't tell me what's going on, right? There's a difference. It says, okay, if I took this part out, you wouldn't be able to talk anymore. You leave it in, you can talk, but I don't know still how you can talk. I mean, what's going on inside the circuit, right? So that's what we're trying to do. A lot of our work here is understanding these topics. So what circuits underlie those actions and how do they work? An analogy in physics is like, you have a magnet, like a large magnet, and it has magnetism and it can stick to things. What's going on, right? So it took a long time to figure that out. It took a few hundred years to figure that out. And we know it's atomic spins. That's the theory we know. So it's the analogy in the brain is like we know these large things like counting we can do, but what's going on is these little circuits of neurons. They're like the atomic spins, okay? But what are the rules that govern how they do things? Now, we can look at things at different levels. You've had a taste already of this. We can look at the very microscopic level of synapses. We can look at mesoscopic levels of populations of a few cells. Or you can look at whole brains or columns of neurons. So you can pick and choose. Different problems might be better answered at one scale versus another scale. That's the art of doing science, right? You can't do everything at the same time. You have to pick and choose. The field that I'm describing is called neurophysics, and it's doing brain theory basically and understanding these spikes and how they arise and what they're used for and how they compute. But neurophysics also relies on these other areas like imaging that you've seen, interfacing neurons to materials, which is very useful these days for brain-machine interfaces, like you just saw in that video that Richard showed at the end. There's an interface reading the electrical activity off of that paralyzed person's brain. Magnetic field measurements, electric field measurements, brain volume measurements, PET scanners, all these things are part of neurophysics as well, okay? And it's a highly interdisciplinary area. We're training, this is the field of physics that's the most important for understanding this, and this one as well. Information theory, electricity, magnetism, biophysics, and computation, how to do computations on computers. And it's part of the larger area of biological physics. So that's where we sit. Okay, now we're looking for the neural code. We're trying to crack that code. Do we know other codes in biology or elsewhere that have been cracked? Well, we know the genetic code that was cracked by Watson and Crick, right? Many, many years ago. So we know that you assemble sequences of letters, which are proteins, and if you have 3 in a row, and you write them like this, well, this will code for this kind of amino acid, and this has been worked out in great detail. So we know exactly how the genetic code works and can code for various proteins that build up your whole body, right? But we don't have that for the brain. We're just like little bits and pieces, right? We know hippocampus is important for memory. Here's an example of the kind of problem we face. Jean-Claude was saying you have a neuron that receives about 30,000 inputs from other cells. Well, this is the typical problem we face. This is one neuron, and I only drew 1, 2, 3 inputs up here, and they have an E in front, which means that they excite. They will excite this neuron. But there's this one input that inhibits this neuron. And what they do is they do a number on the voltage of this cell. A cell normally has minus 60 millivolts, roughly. And these inputs move that voltage up and down. And when it goes high enough, you get a spike. And that propagates to other cells. They hear about it, right? So here, for example, is a plot of voltage versus time. And then you say, OK, let's say it got an input from this neuron, whatever, up here, at this time. So that produces a little blip up. But then it got this little input from this neuron a bit later, and that brings it down. And none of these were high enough to reach this dashed line, so there was no spike. All that's happening kind of below the radar. Okay, that's just 2 inputs, one up, one down. Here there's like 3 in a row coming down this input here. This input has bang, 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 and the voltage goes up, 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 and up. Then you get a spike. So that's called summation. Here you have 2 inputs on this branch and this branch. They don't add up. But then if they happen at the same time, they do add up. And you can throw in an inhibition at the same time here, and then they won't add up and pass the threshold. So, and then put this in like with 30,000, okay, then you kind of get lost, right? But that's what every neuron in your brain is doing. So we're trying to figure out, what is this all for? Why make it so complicated, right? Why can't we just do this and go home and do something else, right? But, so that's the question. So here's an example of the kind of problem. This is called gain control. And we plot like the activity of the cell, and then we count the number of these spikes as a function of time. 10 per second, for example, as a function of the strength of the input. And sometimes you get a straight line like that. But under some conditions, the straight line moves over to here. That's what happens when your eye goes from dark to light. You'll get this shift. But sometimes it's the slope that changes. Sometimes you get something from here to here. That's called divisive gain control instead of subtractive gain control. Sometimes you get something that's like that. And it depends on the details. And now, that's the brain. That's certain cells in your brain, certain cells in your retina. They do these kinds of transformations, these input-output transformations. We know a circuit that divides in the brain of a fish that we've studied here in great detail. This is the circuit that actually produces this change of slope. It's this complicated circuit. If you're an electronic engineer, you know how to do gain control. It's very simple. I mean, we all learned this at first year physics, right? But that's how the brain does it. It's a bit more complicated, okay? So we're trying to work out different aspects of these computations that the brain does and trying to figure out how the system is designed. So for example, from the outside world, you'll have visual stimuli that propagate forward up to different layers, like Richard was showing, going through different layers of the brain. But the thing that wasn't so clear from that plot is that there's a lot of traffic going in the other direction. There's a lot of stuff happening from upper brain coming back down. So in those 300 layers that Richard was showing, it was all going one way. That's how deep learning works. You've heard of deep learning maybe in AI. That's how deep learning works. But the real brain has a lot of stuff going the other way. So we're trying to figure out what's that design about. Okay, that's called feedback. Roles of feedback, various have been proposed in the literature. You can get switching between patterns. You can get brain rhythms. You can stimulate. You can estimate what's the stimulus coming. You can learn. You can focus attention. That's the role of this traffic coming back the other way. Those are computations that have been figured out as part of the neural code so far. So this guy, a great inspiration to many of us, he figured out that you can be looking somewhere in the middle here, but actually you're paying attention to here. So your eyes are looking there, but you can actually read off the letters here because you're paying attention to the letters here. Or we can be having a conversation with somebody and we're actually paying attention to what's going on there, right? It's called covert attention, right? So he figured this out, and that's a problem of feedback and gain control. That's exactly a kind of feedback and gain control, is how do you, from the top down of your brain, you can go and direct the attention somewhere else, okay? That was one of his pastimes. He worked out lots of things about the auditory system, about vision, but in physics, he's known for the one who discovered conservation of energy. Okay, so that's a big deal. That was Helmholtz, okay? So why is understanding feedback important? So now I'm going to go more towards the clinical stuff. So here's an example. It's a bit technical, okay? But it helps us predict this feedback. And we're trying to figure out more about this. So let's say I have a motor command. I'm gonna speak. So this motor command goes to my vocal cords. And I produce a sound. And that goes to my ear. And then that goes into my brain. But what the brain does at the same time is it sends an internal signal saying, watch out. I just sent a message to the mouth to say something. So the ear, you better get ready because you're supposed to expect this thing that I just sent to the mouth. And if it's not what you expect, then there's something weird going on, and that may be interesting. And that's this feedback pathway here. It's called corollary discharge. And then you check the discrepancy between what you expect. No discrepancy, you'll actually attenuate things. You expect it so you don't pay as much attention to it. Okay? So that's internal feedback. Okay, so we try and work out these circuits. This is actually, I wrote up there, tinnitus. This is actually one of the theories. When this circuit is messed up, it can lead to tinnitus. Okay, that's one current hypothesis. Okay, here's another one, a big one, and I collaborate with people on pain research. In your spinal cord, you collect all the information from your pain sensors and you send that up to the brain. So pain signals in your skin go up to the brain. But the brain also has this other pathway, it's called feedback, comes back from the brain down to your spinal cord, and it actually acts as a little gate here. And it says, let the signal go through, or don't, or modulate it in some way. Chronic pain is a problem with that thing right there, with that circuit, because this nerve has been damaged, and this thing thinks that it's always, should always be told, that the brain should always be told there's this thing that's damaged. But after a while, the brain knows, and we need to quiet down that signal. So this gating theory was proposed by Melzack, back in the 60s. We're still trying to figure out exactly how it works, because it turns out that the circuitry right here is a mess. There's thousands of different neurons in all kinds of layers, and we're trying to work that out. What's the circuitry? How does it work? Okay. Brains have circuitries that are mapped out these days in great detail, and we try to figure out what this is, what's it doing, different parts of the brain. That's called a connectome. So lots of research going on in that. But we're not the first ones to do this. So back in the 1300s, this chap, Avicenna, philosopher, scientist in Persia, was very interested in the circuitry of the brain between the eyes and the ears, and Da Vinci also was working this out. So we are, standing on the shoulders of giants to do a lot of our work. Okay, so very important. We study circuits, in simplified animals like fish, and we try and work out the details of these circuits, and we can cut or temporarily paralyze different pathways in the circuitry and see what happens. Okay, but the point is that we study simple animals to figure out some computations, because it's too complicated in humans, right? So we try and figure out, for example, how these fish communicate with one another. These are two fish. When they're next to each other, they hear this wobbling sound because they each have a frequency, and they cause a beat frequency, and they can tell who's around. If a third person shows up, well, it produces this little waveform. And this is the basis of what our auditory system is doing as well. But it's a much simpler circuit than our auditory system. So we study this one instead. Richard and I actually put out a paper a few years ago on how frogs count. So frogs count. They have cells in the brain that count how many calls are coming from other frogs. Right? They're in a swamp. There's calls all over the place. But then they have these cells that will fire if there's 8 calls coming from that direction. That's someone they're interested in, right? And it's amazing, right? So we try and work out, okay, and is this the basis of how we count? I probably won't know in my lifetime, but maybe this work will help understand. This is the circuit that we propose to do this, so we won't go into that any detail. But of course, there's big questions like consciousness that we like to philosophize about, and so this is controversy about the mind-brain problem. Can consciousness even be investigated scientifically? We look for physical correlates of consciousness. And then there's various theories like classical physics is enough, or quantum physics is necessary, or all physics is not enough anyways. This dualism. Are we machines? All these questions come up. And my view, and that of many others, is that we are kind of this emergent computational organism. We put together lots of simple parts, but when you put a lot of these together, they produce things you would never dreamed of before. The sum of the parts is much greater than the simple parts. That's what nonlinear physics studies, is exactly this problem. So part of it is that we don't actually know all physics yet. We need to study physics at the same time as the brain. As this guy pointed out, he studied quantum mechanics many years ago and wrote this amazing book. And he came up with this idea that the activity of the mind is kind of random and deterministic together. And this guy, who you've seen movies about, Alan Turing, also came up with made predictions about what makes the brain work, and a machine cannot do anything new. But what about a machine with some randomness in it? And that's us. We're like a machine with lots of randomness in it. Well, maybe that does new things. So him and Schrödinger together and lots of other people are kind of studying the noise and the randomness in the brain along with some kind of new physics to understand what's going on in the brain. So in practice, while waiting for answers to these big questions, back at the ranch, we can study things like coding in disease, like practical problems, and I'll end with that. Here's what happens inside one of your wires, one of your axons, when you've had a stroke or trauma. So it's normally a pipe, and it's supposed to be a pipe here, but here there's been a little detached part. It kind of blebs out. We call it a bleb. And this blebbing changes the properties of these channels, these ionic channels that let the currents go in and out. So trauma will do that. So Kathy Morris, who works here, we've collaborated with her. She worked at the Civic all her career. She studied this in great detail. And we've been working with her to try and figure out what this does. So for example, we look at things like voltage versus time in a healthy axon. Well, that looks awfully boring. Nothing happens. That's what an axon is supposed to do when there's nothing going through it. It's not supposed to do anything. It's supposed to only transfer information going through it if something goes in. But this damaged axon, what it does is it actually goes fire, 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 stop, fire, 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 stop, fire, fire, stop. And it does this for days and months, and you have chronic pain. It's not supposed to be doing that. If you get a concussion, it'll start doing that or this. These are not good. So we try and figure out from our modeling what's going on and how can you repair it. We know, for example, that if you cool the system down, you can go back to this flat line again, which is good. So you can study how temperature can actually help, relieve trauma and so on. So that, in a nutshell, is the kind of work we do in the brain and mind here, where we study circuits in simplified animals to try and understand the principles of coding. It's important for big questions like consciousness and so on, we hope. It may have nothing to do, I don't know. But in the meantime, I think it's very important for curing disease. And with that, we'll stop there and take questions. Thank you. So, are there any questions? Or comments? Or
when you talk about cooling in the sense of a concussion, what do you mean by that? How do you cool?
Well, I can put an ice pack.
Oh, so it's as simple as that?
As simple as that. Or if you're like, what's the race car driver, Michael Schumacher, you cool his brain down. I mean, it didn't help. He was in induced coma, right? But you try and manage the damage with cooling. With cooling, you kind of lower the activity of the neuron, and you can suppress abnormal firing. That's the idea.
So cooling, putting ice on, and it lowers the activity. Is that
exactly.
What
yeah.
Okay.
Or they have other ways of cooling the blood. Yeah.
Sorry, what's cooling the blood?
They have other ways of cooling the blood, I think,
in where?
Chronically, so they keep the person with cool blood for a long time.
Thank you.
Yeah. Until things repair properly.
Oops, sorry. Thanks. Sorry.
So I'm studying how implicit stimuli actually make our systems more efficient in terms of memory. And I was just wondering, I really like the feedback loop, and if whether or not you've demonstrated kind of how hidden stimuli or unconscious stimuli affect that in like simple computational models or maybe like simple animal models. Because like for us, it's that we take it in and we discard it, but then it actually comes back and it makes us faster or it makes us more accurate. But I'm just wondering if that has been seen in your models.
So I haven't studied implicit memory directly. Have you? No? No. 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 So I mean, it gets beyond the complexity of the hippocampus, where the stuff is stored to long-term memory in the first place, and then where that long-term memory sits is kind of in cortex, and that feedbacks to everywhere. It's a very, very tough problem. So at that level, I think we're still far from making headway, unless you can formulate an implicit memory problem with a sea slug or with a very simple creature. Maybe they can get at that.
Thank you.
Your question was about whether computationally we've studied this, and I don't know of any studies in that direction. Yeah.
Hi, I have a question about the AI, the speed that it's going. Are we as human beings, should we worry about it? You know, the alpha system that they can play the Go game is winning the professionals by tons amount, you know. So are we going to be replaced someday soon? I thought, you know, it'd be beyond me way down the road, but it's coming fast.
I'm not an AI expert. I'm using AI to understand the brain, but I do work with AI experts. And well, one should always be careful in every type of technological developments that we do, and my take is that we should try to be as careful as we can, but we don't really know how things are going to go. And it's true, it's very impressive, and that's why there's a lot of work right now of people working in ethics and ethics of artificial intelligence. Some people in artificial intelligence will tell you that there's nothing to worry, it's still very limited, it can only do so much. I find that the paces at which things have been going would speak against this. So I'm more an advocate of being careful in the way things are developed. Yeah.
Thank you very much. It's been very interesting. I like some questions answered about HM. Was there a time lag between when the experiments were done? Was he older? Was he younger? Was there variables that could have affected this, or was it a straight line between what happened to him and his ability or losses of memory?
On that, so yeah, that's a good question. And so Dr. Milner has studied HM for, I think, more than 30 years. So the level of experiments that were carried out were increasingly sophisticated. As far as I understand, and again, I'm not an expert on cognitive neuroscience, but I don't think that his condition has evolved. Like the primary features of the neurological or the cognitive deficits that HM experienced evolve themselves in any significant way over time. You may want to comment on this. No, I'm looking at you. Yes? No? Thank you. I got them. Any other questions?
Thank you. Very interesting. I'm currently going through neurofeedback for a concussion. Can anybody make some comments on your beliefs or feelings on it?
I don't know enough about neurofeedback and concussion. I think there are a lot of interventions on different concussion that people don't have a lot of evidence for. So I hate to say anything definitive about it. Just I don't know the evidence for that type of intervention.
Any of you gentlemen?
I'm gonna take the lead of Heidi on that one.
I think it has a lot of promise because we're always trying to regulate what we're doing through some kind of internal feedback mechanisms,
Well, nice to hear, because I've been spending a whole lot of money on it. But it has helped me memory. It just hasn't helped me with balance yet. So we'll see. Thank you.
In terms of the example of the cooling that helps with concussion, is it only cooling, or is it also applied to things like having simpler tasks that could kind of normalize that versus complex tasks that could possibly trigger those?
So I would say, yeah, that's an excellent question. I mean, actually, Heidi and I did some research on this.
Microphone for the folks who are online.
Yes, thank you. So from behavioral standpoint, it's clear that a lot of your deficits following concussion do manifest when you're trying to do complicated tasks, right? That's why they'll put a hockey player, they'll say good to go, and then they're in an actual situation and everything just gets jammed up because there's too much happening. This is a very simplified picture. I would have to have a way to model the complexity of a task. Somehow there have to be many things going on that are being controlled, and then see, but I think it'd be easy to show if you have a simple task, you can regulate it better than if you have too many things at the same time. Because I think the cognitive load means that some parts of the feedback are not working properly. The signals just get jammed. But to get a really useful prediction out of that, I can just anticipate that answer if I wire up the model the right way, But to get something very useful with that, you have to really have a close interaction with an experimentalist to set up the question and to make a prediction that can be verified experimentally. Go check out this thing, But with fMRI, I think they're doing that. They're looking at the connectivities of different parts of the brain, and that's kind of messed up when your concussion isn't healed yet, right? As far as I know.
Yeah. All right. So I think we'll end it here. So we're going to stick around outside. So if you have maybe you want to ask them some more questions. So thank you all again for coming. I think so. Probably you're a little bit confused, right? Because so you're all bombarded with information from different, yet complementary, but still distinct fields. And maybe it gives the impression that it's a bit messy. And the truth of the matter is, this is a good analogy of how we study brain. It's hard, and it is messy. And as scientists, oftentimes we get attracted by messy things. And the brain it's hard. And what we do, it's hard to approach. How do you approach these complicated problems? Are we dealing with a 1965 Lada that we're just trying to tinker with? Or this is amazing Formula One that we're trying to take apart? So this is what we kind of do in our labs. We tinker with things. And with the brain, it's in neuroscience in general, I think it's a good sort of humbling reminder that we have a puzzle in front of us, but we don't even know how many pieces we have. Do we? And in terms of how, like, our understanding of this puzzle, how close or how far we are, it's very difficult. And I'm going to use that analogy to push a little bit for the use of basic science in neuroscience research. We don't know how many pieces we have. Do we have 500 or do we have 3,000 left? We just don't know. And so we need to be able to be driven by what we call scientist curiosity-driven research, which may or may not be used to treat diseases at some level, but we're so in the dark of how to approach this that we need to put a lot of value in the types of basic questions that we ask. Like, for instance, Andre was talking about, how do frogs count? And you're like, okay, why do we care about how frogs count, right? But it turns out that by these understanding these simple mechanisms, these these simple problems in more simple organism tells you a lot about how the brain works, and that's the only way we can actually figure it out how to fix it when things go wrong. So with that note, so we're going to end here. I want to thank Richard and especially Emerson, who is our really, these guys are the ones who do the hard problems. And we hope we've been able, if not to confuse you, perhaps give you a little bit of enthusiasm for the type of research that's carried out at the university. And if you're interested to support it in any way, shape, or form, you can come outside and we can give you information and things like that. So thank you all for coming, and especially on a Friday evening, and see you next year.